right. So this is now part 1B of our unit, peptides and proteins. And what we're going to focus on in this module is asking the question of how were amino acids formed. Okay, so let's take a closer look. So again, here's our outline. We're now focusing on part number 2, how were amino acids formed. Okay, so this is a question that was asked by two scientists by the name of Urey and Miller. And, the question they're asking is it a coincidence that amino acids are what they are? Is there a way to form them in a particular way using very basic building blocks that may have been around in the universe after, for example, the Big Bang theory, the Big Bang? So, the sun, as you know, is comprised of hydrogen. So hydrogen is the simplest and the most reduced form. The most reduced form of carbon is methane. And methane is the simplest reduced form, CH4. The most reduced form of nitrogen and simplest form would be ammonia. And if we are going to make amino acids, we need to have some sort of oxidizing agent, some way to add oxygen, and water would seem like a natural, sort of a natural molecule to think about in this type of experiment. So it turns out if you actually mix hydrogen, methane, ammonia, and water together, mix them in a vesicle for some time, they're not going to react. Okay, they're very stable molecules. There's really nothing driving that reaction. However, if you think of, the environment and you think of the universe or even think of planets, there would have been moments of electrical discharge that is high energy that may have arisen as a result of things like lightning, high kinetic energy from meteorites moving through the solar system and impacting and touching each other. So lots of opportunities for high energy collisions, electric discharges. And the question was, well, if we take these basic building blocks together, will we actually construct the backbone of amino acids? So this was the very question that Urey and Miller sought to seek when they were looking at this experiment. And this experiment was done in the, around the early 1800s. So this was done a very long time ago. And essentially what they had done was they had taken this contraption, which has compressed gases inside, okay? So what they had inside was they had things like methane, they had things like hydrogen, the ammonia that we were talking about, and then this boiling water supplied water, H2O, of course. So when they added these gases to the lines, you can see that they would be compressed, they would go through these lines, and then the system here was their wires for electrical current, again, used to mimic an electrical discharge. And then any products that were formed would be condensed and this cooled down into this trap. And then any amino acids or any byproducts that would be formed would be contained in this area here. And then this would kind of recycle and go back and forth. So any unused materials would go back and forth into that area. So what they noticed was that when they did this experiment and did this multiple times with that electrical discharge, and again, just emphasizing that this comes from a primitive environment, they observed that they could generate hydrogen cyanide plus an aldehyde. In this case, the simplest aldehyde, formaldehyde. So these were formed as gases and they would keep going back and forth. And then eventually these would react with ammonia to give us the core backbone of an amino acid. So here you have an ammonia, NH2CH2CN, and when this reacts with water, this cyano group becomes hydrolyzed, and this becomes the backbone, or this becomes our amino acid as shown. So let's take a look at or summarize some of those findings from that experiment. Okay, so what they noted was that during the Urey-Miller experiment, they noted that they got rapid initial formation of hydrogen cyanide and aldehydes. The most prominent one was formaldehyde, which is the simplest aldehyde, and this eventually would lead to glycine. But they also observed other aldehydes too. Okay, so you can imagine another R group here. This could be a methyl group or other more complex functional side chains. And this would give rise to our amino acids. And as you know, amino acids are characterized by this chiral carbon, which has 4 different substituents and different R groups. So there's a lot of evidence suggesting that depending on the aldehydes that you get, these would make the amino acids. Later on, a German chemist by the name of Adolf Strecker developed in 1850 a synthesis to give racemic amino acids. Okay, and so using the sort of the experiments done by Urey-Miller, he and his team looked at taking aldehydes plus hydrogen cyanide with ammonia in an aqueous environment and wondered whether they could actually generate a large-scale amount of these amino acids. So what we're going to do is we're going to take a close look at this mechanism known as the Strecker synthesis, and this will be our first sort of big mechanism. And what this is going to look at is taking some type of aldehyde, and remember we have generated hydrogen cyanide from that experiment, and we're going to see how these molecules can come together to generate amino acids. Okay, so here you have an aldehyde and hydrogen cyanide. In order for these two to react together, they're both actually, they're both not reactive to one another. However, you can think of hydrogen cyanide as being an acid, and you can think of the aldehyde as participating as a base. So if this is the case, you now have electrons from the oxygen picking up the proton from hydrogen cyanide, like I've shown there. And then what's going to happen next is you're going to now have a situation where you have an activated aldehyde and a cyanide anion. Okay, now remember that the ingredients was not only the aldehyde plus hydrogen cyanide, but we also had ammonia present. This was our source of nitrogen for the formation of those amino acids. So in order for that to occur, or for that to participate and be used in the reaction, the ammonia actually is a fairly good nucleophile. Okay, so you have electrons on the nitrogen. This aldehyde here that has now been protonated and has a positive oxygen is also known as an oxonium ion. It's high in energy, it's activated, and it's an excellent electrophile. Okay, so it's seeking electrons. This nitrogen here is a very good nucleophile. So what's going to happen is you're going to get attack of the ammonia to the aldehyde. And when this happens, you're going to now generate a species that looks like this. Your nitrogen now is positively charged because it has 4 bonds. And remember, the pK of the ammonia here is around 10 or so. And we did form cyanide anion. So what can happen is that a base can come and remove that proton to make this compound neutral. Once that's done, we take another molecule of hydrogen cyanide, and this OH molecule is now going to pick up a proton to become positively charged again, and we are going to generate a good leaving group. Okay, so at this stage here, we need to find a way to add another carbon to this molecule, okay? Because at the end of the day, and maybe I'll draw it over here in this corner, we are generating amino acids, and amino acids have a carbon here, but we need to add another carbon over here. So the carbon that needs to be added is going to come from the cyanide anion. However, if we have our cyanide anion here, there's no good way for it to come and attack this molecule. So what we're going to do is we're going to take advantage of the nitrogen lone pairs, and that's going to help push out the water. And what we're going to have now is an iminium ion, which is now very, very electrophilic. So I'll just write that down, very electrophilic. And that means it is also seeking a nucleophile. And now at this stage here, the cyanide anion can come and react like I'm showing you there. And what you can see if you compare the structure here that I've drawn and compare it to the one in the lower right-hand corner, you can see the overall backbone is actually the same. It's actually, you have a nitrogen, carbon, carbon. Here you have a nitrogen, carbon, carbon, R group, and then hydrogen, R group, hydrogen. So what we need to do now is we need to take this nitro group and we need to hydrolyze it. Okay, so this is where our water comes into play. And what we're going to do is we're going to assume that because we are now in an aqueous environment, we're going to have access to, well, of course, water, but we're also going to have access to a conjugate acid of a base, so a good proton donor here. So water is going to come and it's going to attack the cyanonitrile group, and then this is going to come and attack, and the triple bond will pick up a proton. And what we are going to have is the addition of one water molecule to this construct here. Now, at this stage here, what we're going to do is take a base, remove this proton, form a double bond, and then this is going to come pick up a proton from the conjugate acid of the base that actually just picked up the proton. And then now we're left with this compound right here, and now water can come react at this center here. When it reacts at the center, you now have OH, you have NH2, and now you're going to have OH like this, OH2. So now we've added two oxygens across this nitro group. And then finally, we need to deprotonate one of those oxygens. And that's going to look like this. And what we need to do, I hope you can see from here, is we need to lose the NH2 group and form a double bond between one of the oxygens and the carbon. So in order to do that, we need to make this NH2 a very good leaving group. So we need to protonate that. So that can be done with a generic acid. And then at this stage here, we can take electrons from an oxygen that will come down and then help kick off the ammonia molecule. And then finally, at this stage here, all we need to do is a proton transfer to remove that proton to give us our amino acid. So this is known as the Strecker synthesis. It's a well-known reaction. It's actually used industrially to make a wide range of amino acids, and it's used today for various companies to synthesize these as chemical precursors for researchers. So just to summarize, the results of the Urey-Miller experiment, so as they were making this compound, it wasn't a super clean reaction. They got some insoluble material, also likely a cyanide aldehyde polymer. 10 to 15% of the carbon had been converted to organic compounds, including amino acids. Not surprisingly, glycine was found to be the most abundant amino acid, the simplest amino acid, right? So it comes from formaldehyde. But of the 20 amino acids that we know in modern cells, 12 of the other proteinogenic amino acids were formed. And that's pretty impressive when you're considering an apparatus that was done that probably didn't meet the standards of safety and basically took these very simple compressed gases and heated them up and added electrical discharges to them. And as you know, these were called alpha amino acids. Other examples of amino acids that can be made too that were also detected were things that are not necessarily used for proteins or peptides, but other things. So for example, this is known as GABA aminobutyric acid. It's a gamma amino acid. This is actually a very important neurotransmitter that's used biologically. So you may have heard of this one before, but you can see it's actually just an elongated amino acid. The amino acids used for protein synthesis are always L. We talked about that in the last unit. At the alpha stereocenter, except for glycine, which is S in configuration. D amino acids, okay? So we talked about L amino acids, those are the natural ones. D amino acids do actually occur naturally. They are quite rare, and they can be sometimes found in non-ribosomal peptides. And then the last thing I just want to indicate is some evidence showing that what people have done in lab with the Urey-Miller experiment, then optimizing it with the Strecker synthesis, is that some amino acids have actually been detected in space. So in 1969, in Australia, a meteorite actually fell down on the country, known as a Murchison meteorite. And at the time, researchers immediately went and started to take the surface of the meteorite and were wondering what was on that surface. So what was really interesting was that, well, if this meteorite was in an environment that had these natural gases that we're talking about, right, like hydrogen, ammonia, methane, maybe even water, and presumably over time it would have been exposed to brief moments of electrical discharge, right? So what was on the surface? So researchers by mass spectrometry actually noted that, well, on the cell surface, not the cell surface, the surface of the rock, there were alkanes, so very simple alkanes, but they also found glycine, right? So remember, glycines are simplest amino acid. They also found alanine. Alanine is the next simplest amino acid, where the R group is equal to a CH3. And then they also found other amino acids like glutamic acid. And on the surface of these meteorites, they found not only these amino acids, but they found both L and D amino acids. Okay, and you might be thinking, well, we know we live in an L amino acid world, and this is going to segue to the next section. But on the meteorite, yes, L and D amino acids were present, but there was a slight enantiomeric excess of L. And I want us to think about that a little bit, because why is that? Why was that? And why do we live in a world full of L amino acids? Okay, that's it for this session.